Hi gang, welcome to the second video for Chapter 6, Microbial Nutrition and Growth, Physical Requirements for Nutrition and Growth. So in the previous video, we went over the chemical requirements or the nutritional factors for growth, and those were these atoms, carbon, nitrogen, sulfur, and phosphorus, and also hydrogen should be in there. Trace elements like magnesium, oxygen, molecular oxygen, and organic growth factors. So organic, just remember, has a carbon and hydrogen atom in it if you are an organic anything. So this video we're going to talk about physical requirements that microbes need to grow. So they need a certain temperature range. Certain microbes need certain temperatures. Other microbes will need different levels of pH. We kind of went over this in the chemical requirements. If you are an aerobic organism, you need oxygen. If you are an anaerobic microbe, then oxygen is toxic to you. Microbes grow at different hydrostatic pressures and osmotic pressures, and we'll talk about all those in the physical requirements today. This is just a review of the chemical requirements for nutrition and growth of microbes. So here we are, all organisms, and they need an energy source. We all do, whether we're a tiny microbe or a human being. The energy source could be a chemical energy source or a light energy source. If you use chemicals as your energy source, you are a chemotroph. If you use light as your energy source, you are a phototroph. But whether you are a chemotroph or a phototroph, you need food source. That's your carbon source. If you're a chemotroph and your carbon source is an organic compound like glucose, C6H12O6, then you are a chemoheterotroph. And then what is your final electron acceptor when you're breaking down glucose? If your final electron is oxygen, your final electron acceptor is oxygen, then you are most likely an animal. Could be most fungi, protozoa, and some bacteria. If your final electron acceptor in the electron transport chain is not molecular oxygen, and then it could be some organic compound, then you would be doing fermentation. Or it could be an inorganic compound as your final electron acceptor. So if you are a chemotroph and your carbon source is carbon dioxide, then you are called a chemoautotroph. So let's go back over to this side. If your energy source is light, then you're a phototroph. If your carbon source is an organic compound, then you're a photoheterotroph. If your carbon source is carbon dioxide, then you are a photoautotroph. So now we'll talk about the physical requirements for growth. But before we do that, we have to do a little chemistry from Bio 201. If you notice this is H2O, that is water. So the big red ball is hydrogen, is oxygen. And then these guys are hydrogen atoms. And then here's another water molecule. Here's another water molecule, another one, and another one. So there are 1, 2, 3, 4, 5 water molecules in this cartoon. And they're all attracted to each other through these things called hydrogen bonds. So this symbol is a partially negative. So partially negative oxygen will hydrogen bond. Hydrogen bonds are symbolized by these dots or dashes. So a partially negative oxygen of one water molecule will hydrogen bond to the partially positive hydrogen atom of another water molecule. And then they show it down here too. So that's how you probably introduced to hydrogen bonds. Hydrogen bonds not only form between water molecules, they can form between any electronegative oxygen and an electronegative, partially negative oxygen and a partially positive hydrogen atom. And then here's these dashed lines that show this is a hydrogen bond. So this, remember, this is partially negative, and this hydrogen is partially positive, so they'll form a hydrogen bond. Notice that this oxygen that's partially negative is connected to a carbon. This hydrogen, which is partially positive, is connected to a nitrogen atom. And then this hydrogen that's connected to a nitrogen atom is also partially positive. And here's another oxygen that's connected to a carbon atom. And this oxygen is partially negative. And then the dashed lines are hydrogen bonds. This just shows that you can have two hydrogens, or you can have a hydrogen bond between a nitrogen atom of one molecule and a hydrogen atom of another molecule bound to nitrogen. So hydrogen bonds, they come in many flavors. So hydrogen bonds and proteins. So probably know proteins are made from amino acids. Here's amino acid 1 in this cartoon, amino acid 2. And amino acids, they contain, here's a central carbon with this thing called an R group, and this R group is different in every amino acid. Amino acids have an amino end, that's N, a nitrogen with 2 hydrogens, and they also have a carboxyl end, which is a carbon double bonded to an oxygen and a carbon single bond to an oxygen bound to a hydrogen. And here's amino acid 2. And to build a protein, you need to hook up your amino acids, and you do that by making this thing called a peptide bond. When you make a peptide bond between 2 amino acids, you remove a water molecule. And that's what this orange coloring in here is. When you hook amino acid 1 to amino acid 2, you remove the 2 H's here and here, and an oxygen, that's H2O. And then you form this thing called a peptide bond. And the peptide bond is between the carbon in the carboxyl terminus of amino acid 1 and the nitrogen in the amino terminus of amino acid 2. And then you have this water molecule that was removed to form the peptide bond. And here's just these are cartoons of some of the R groups that you could potentially have on an amino acid. So for example, the amino acid glycine, the R group is just a single hydrogen atom. In alanine, the R group is just a methyl group. A more interesting one, in aspartic acid, the R group is a little more complicated, but it's got this negatively charged oxygen. So the key is this negatively charged oxygen could form a hydrogen bond with a partially positive hydrogen somewhere in the protein. Same with this negatively charged oxygen. So these, in other words, all these cartoons are R groups of the different amino acids. So hydrogen bonds in proteins. So this is a stylized protein molecule. It just shows the different hydrogen bonds in a protein molecule. So you could have a hydrogen bond between the atoms of 2 peptide bonds. So in this cartoon, the orange is the hydrogen bonds. So this would be an atom involved in a peptide bond and another atom involved in a peptide bond. In your protein, you could form a hydrogen bond between the atoms of a peptide bond and an amino acid side chain. And we saw all those different amino acid side chains. You could also form a hydrogen bond between 2 amino acid side chains. I should tell you, the side chains are also called R groups. And in the previous slide, we were calling the side chains R groups. But the point is, proteins and their shapes are held together by these hydrogen bonds. These hydrogen bonds can be disrupted by high temperatures and acid conditions. And if you're a microbe and you have an enzyme that is a protein and you need that enzyme to live and the temperature gets too hot, then these hydrogen bonds will fall apart, the shape of your protein will fall apart, your enzyme won't function, and you will die. So, proteins, which are often enzymes, proteins catalyze chemical reactions that are needed for growth, for microbes to grow. They require this specific 3D physical shape. So, for example, this would be the 3D physical shape of a protein in cartoon form. They need this shape to function, and the protein is held together by hydrogen bonds, which we just talked about. Hydrogen bonds can break if you increase the temperature, because remember, these bonds, they're not that strong. So if you heat them up, they fall apart. Lipids are also temperature sensitive, and they can become too fluid at high temperatures or too brittle at low temperatures. And then remember, the plasma membrane is a phospholipid bilayer. That's not good if you're a microbe to have your plasma membrane fall apart. So let's take a look at this diagram. So in A, it's got the growth rate of a microbe here. Here's the minimum growth rate. And then it's got, on the x-axis, it's got the temperature. So, and then it's got this dashed line called optimum. So you can see the growth rate is the highest right here at that microbe's optimal temperature to grow. This would be the, it can still grow at these higher temperatures, can still even be growing a little bit here, but this microbe has an optimal growth rate at this temperature, and it's not shown on the y-axis, but down here it shows this is a microbe, doesn't say what the microbe is, at 22 degrees C. You can see the growth here and here. At 30 degrees C, you can see growth here. And then at 37 degrees C, you see the most growth. So you've got growth here, here you've got growth all out in here. All of these little circles are colonies of microbes. So there's minimal, maximal, and optimal growth temperatures for each microbe. All right, so this is a cartoon. It's got the growth rate on the y-axis again, the temperature on the x-axis. And you can see these different categories of microbes growing at their optimal temperature. So for these guys, psychrophiles, for example, you take the optimum growth rate, which is down here, and then you go, I don't know, it's about 10 degrees C. Mesophiles grow at about 37 degrees C, oops, which is our body temperature. So mesophiles will grow in us. And then you have thermophiles, hyperthermophiles, and then I drew in this part of the graph because they didn't have it on this cartoon. But there are microbes called xerophiles, and you would likely find those growing in the desert. So I could even made this peek over here a little bit more, how hot the desert gets. So, for example, if you are a bacterium and you are a mesophilic aerobe, that means you grow in an oxygen-rich environment, and you grow at 37 degrees C, and yeah, requiring oxygen, I said that. If it's a neutral environment, that means the pH is 7. So the psychrophile grows at low temperatures. So there's a psychrophilic alga called Chlamydomonas nivalis growing on a snowbank. And you can see it's kind of, I don't know, what is that, pinkish red? So they grow best at temperatures just below 15 degrees C. They cannot cause human disease because our body temperature is 37 degrees C. Here's the ones that can cause disease, the mesophiles. And I thought this was really cute. It's like a little card about mesophiles, excuse me. And it says up here, most diseases caused by bacteria and viruses. Well, bacteria are mesophiles. Human pathogens are mesophiles. They grow anywhere between 20 and 40 degrees C, but normal body temperatures, like I said, 37 degrees Celsius. The mesophilic organism Helicobacter pylori is responsible for your gastric ulcers if you have them. Okay, next guys, the thermophiles here. They grow best above 45 degrees C. You can find them in compost piles and hot springs. If you are a microbe and you are growing in a hot spring, you are generally a thermophile. But if you can grow above from minus 80 to 121 degrees C, so above boiling, then you are a hyperthermophile. So you're over here. Microbes that can survive boiling are generally called hyperthermophiles. You can have archaea that live in deep ocean boiling mineral hot springs, which is this picture. They can't cause disease in us because we don't live at those high temperatures. And this is a photograph from Yellowstone National Park that shows where microbes are growing, and they give off these different colors, which is really pretty. Physical requirements for growth. Microbes need a certain pH. It's the acidity. This is an example of an acidophilic microbe that lives in an acid mine drainage. So this color in this mine drainage is due to the acidophilic microbe that lives in it. So remember, pH is a measure of the negative log of the concentration of hydrogen, free hydrogen ion. Below 7 is acidic, above 7 is alkalinic. Most bacteria and protozoa are neutrophiles. They like a neutral pH, and they can grow between 6.5 and 7.5 with an average of 7.0, which is a neutral pH. That's kind of the range of our pH. So our blood pH is 7.4, which is in that range. Some bacteria, many fungi are acidophiles. They like acid environments. Normal resident strains of the acidophilic bacteria live in the vagina. And acidophilic organisms are found in yogurt. So if you like to eat yogurt, you'll be eating some delicious acidophilic organisms. If you are an alkalinophile, that means you love a high pH up to 9.5. So Vibrio cholerae, causative agent of cholera, is an alkalinophile. All right, now we're going to more physical requirements for growth, osmotic pressure. Let's look at our cartoons. So osmotic pressure, so osmosis, this movement of water down its concentration gradient across the semi-permeable membrane. So in this cartoon, let's say this is a cell, and then here's this green stuff. This is all the solution that the cell lives in. So, or the cell was put in, and say in a beaker that had a hypotonic solution. So hypotonic, hypo means low. That means that there is low salt or low solute concentration outside the cell in the solution. That means there's a high water concentration. And these are relative values. So the solution on the outside of the cell has a low solute concentration relative to the inside of the cell, which has a high solute concentration. So low on the outside of the cell, low solute, high water. Inside the cell, high solute, low water. That means when you put a cell or a microbe, which could be a single cell, you put a microbe, single-celled microbe, in a hypotonic solution where there's high water on the outside and low water on the inside, water is going to move down its concentration gradient from high to low. The cell will swell and it will burst. In the opposite condition, let's say here's a bacterial cell and it finds itself in a hypertonic solution. That means there's high solute on the outside, low solute on the inside. That means if there's high solute on the outside, that means there's low water on the outside. If there's low solute on the inside, that means there's high water on the inside. So this bacterial cell finds itself in a hypertonic solution. That means water is going to move out of the cell down its concentration gradient from high water to low water, and the cell will shrivel up. So then just here's some definitions. A hypotonic solution, that's the first one we talked about. Condition has a higher concentration of salt inside the cell versus outside the cell. A hypertonic osmotic condition has higher concentration of salt in the solution versus inside the cell. Cells can live in fresh water, or cells that find themselves in fresh water. Fresh water would be a hypotonic environment, gain water and could burst. Bacterial cells, for example, that find themselves in salt water, that's a hypertonic solution, lose water, they shrivel up and die. That's called crenation. And you use this technique to preserve foods. So you have a salty environment, the microbes around that food will die. If you are an obligate halophile, you really like high osmotic pressure and high salt concentration, greater than 9%. So for example, you would find that in the Great Salt Lake. So obligate means that the conditions, the high salt conditions, are necessary for you to grow. If you are a facultative halophile, you can tolerate high osmotic pressure. So for example, Staphylococcus aureus is a facultative halophile. It's a potential pathogen that lives on our skin where it's salty. It's a halophile. It likes our salty skin. If you are a prokaryote and you're known as a halophile, you could require salt concentrations of 15% or greater to live in. Physical requirements for growth continued. A physical requirement would be the hydrostatic pressure. So barophiles, the barometer measures pressure. Barophiles live in water under extreme pressure. So this is a photograph of a deep ocean hot springs.